Okay, so my research is in acute myeloid leukemias, and the most difficult part of the molecular biology are the P53 mutant myeloid leukemias. We have worked for 20 years on activating P53, which is inactivated in many AMLs and many tumors by overexpression of MDM2. So 20 years ago, we started blocking the MDM2 P53 interaction with drugs called nutlins, and we did the first preclinical studies in the first clinical trial. So we had limited success, and we then next combined venetoclax BCL2 inhibition with MDM2 inhibition, and we developed a complex mechanism published in Cancer Cell in 2017, where we show that the combination eliminates, degrades, and inhibits MCL1, which is the main resistance factor to venetoclax. And this was done preclinically and then clinically, and actually the paper came out on the same day that we presented the first data at ASH in San Diego in 2017. The final data was recently published in Blood a few months ago. The next thought was, if P53 is a transcription factor, it gets pumped out of the nucleus by an export mechanism called XPO1. And there is an inhibitor, selinexor, which keeps P53 in the nucleus. And now it can have a much higher transcription activity. So we exploited that and combined it with BCL2 inhibition and MDM2 inhibition. And this is right now probably the best MDM2 inhibitor-based combination therapy. We have a tremendous extension of survival in preclinical models, PDX models derived from patients which are resistant to venetoclax. So these are some thoughts about how to make MDM2 inhibition, that means activation of wild-type P53 in leukemias, more effective. The problem is that when we are doing this, we are selecting out very small clones of P53 mutant leukemias. We have seen that now in 2 different clinical trials. So as early as 4 weeks after we start, we can detect P53 mutant cells that we could not detect before. We mimicked that in the lab. We treated a FLT3 mutant AML cell line with MDM2 inhibitor, and after 4 weeks and 2 and 3 months, we generated P53 mutations. We then found out that a single cell was in our culture system that was not only selected, a mutant cell, which was not only selected but also activated. So this brings us to the P53 mutations in AML. Mutations in P53 are by far the worst prognostic factor that one can have. And there is, until now, no direct way to kill cells with mutant P53. So we thought about different mechanisms. To have active P53, the protein has to be folded in the right conformation. This folding is done through HSP90, heat shock protein 90. And we have now a drug that is very active in that regard, and it's able to block the folding. That means if the protein is not folded correctly, the cells cannot survive. And we are demonstrating that with a drug called PUH71, was developed at Sloan Kettering in New York, but never for this indication. And we published a paper in Blood actually last week, where we are showing all the mechanisms associated with this agent. So it seems to be very active in eliminating mutant P53. Next, we found that mutant P53 is associated with very high levels of MYC. MYC has not been targeted so far. All of attempts to target MYC have failed, but we now have a degrader that we are developing with a company in China. And this degrader indeed eliminates MYC, degrades MYC completely, and is highly active in P53 mutant AML. And finally, we are now working with the first specific drug that activates wild-type p53 in the context of mutant p53. This is developed by a company called PMV Pharma. One of the co-founders is Arne Levin, who discovered p53 over 50 years ago, still very active. And they developed a small molecule that changes the conformation of mutant p53 into wild-type p53, and this results in cell death of leukemic cells carrying this mutation. And we have combined this with MDM2 inhibitors because the newly formed p53 can be inactivated by MDM2. We are blocking that activation, and together with MDM2 inhibition plus this small molecule, we see very nice cell death in leukemias that have this particular p53 mutation. So it was a great pleasure for me to present this data, which is quite complex and very abbreviated, at this conference. I've been associated with Italian leukemia research for over 30 years. I've been many times in Italy. My best friends are in Italy. And this conference now has been spectacular, with a fantastic overview of the latest developments, mostly in the clinic for the treatment of leukemias, lymphomas, myelomas. So it's a great pleasure to be in Rome again, the eternal city, with an ever-evolving science in leukemia research, which is being discussed here. Thank you.